The role of platelets in hemostasis. Following injury to blood vessel, following a vascular injury, what happens to platelets? There are three actions or three functions of platelets. As I told you, adhesion, aggregation and activation you can see. Following injury to blood vessels, the platelets aggregate at the site of injury and seal the opening in the vascular tree. This is the first and important step in prevention of bleeding, which is called temporary hemostasis. So this is the first and important step in prevention of bleeding, which is called temporary hemostasis. Simultaneously, mechanisms of coagulation of blood are also activated that form blood clot. The process is combinedly known as definitive hemostasis. So same thing what I have told earlier, formation of temporary hemostatic blood as well as formation of definitive hemostatic blood is one of the important functions of platelets in hemostasis. So though platelet is essential for temporary hemostasis, it also contributes to blood coagulation and clot retraction. Blood coagulation is also known as definitive hemostasis, and clot retraction is something also known as clot retraction. So clot retraction is a separate thing. It's not a blood coagulation is different, clot retraction is different. Platelet also controls thrombolysis. So blood coagulation, clot retraction and thrombolysis are also the functions of platelets. Even it can be asked as 3 markers, functions of platelets, list out functions of platelets. It can be asked directly. For any course it can be asked, mbbs, bds, bpt, bsc, tech nursing, anything. Okay, so platelet functions is very important. You have to know it properly. So I am not talking individually about platelet functions. As a part of hemostasis, the role of platelets is what I am talking about here. So one of the functions, blood coagulation is there, clot retraction is there, thrombolysis is also controlled by platelets. Immediate hemostasis following vascular injury is achieved by formation of platelet plug. It is also called as platelet thrombus. So platelet thrombus is formed at the site of injury when there is a vascular injury. As platelet thrombus can be washed away under supported by a clot, this is called temporary hemostatic plug. Temporary hemostatic plug formation depends on adhesion, platelet aggregation and release reaction. So coming one by one, what is platelet adhesion? Platelet adhesion. The damage to blood vessel exposes underlying portion of the vessel wall that are normally concealed from the circulatory platelets by intact lining of the endothelium. The platelets have high affinity to adhere to the exposed vascular, which is called as platelet adhesion. Especially exposed collagen, as a part of injury the collagen is exposed, and even there is a factor known as von Willebrand factor in the injured vessel wall attract platelets, as platelets have receptors for them on their cell membrane. The platelets have receptors for the exposed collagen and the von Willebrand factor, so they attract each other and it results in the platelet adhesion. So platelet adhesion is immediately followed by the platelet aggregation at the site of injury. So that's about platelet adhesion. The second one is platelet aggregation. Fibrinogen, glycoprotein 2B and 3A, that's GP, 2B and 3A, and thrombin promote platelet aggregation. So fibrinogen, glycoprotein 2B, 3A and thrombin promote the platelet aggregation. Fibrinogen, glycoprotein 2B, 3A and thrombin promote platelet aggregation. Simultaneously, platelets are activated and activated platelets release the chemicals from their granules. These chemicals, especially ADP and platelet activating factor, that is known as PAF, ADP and PAF further facilitate the platelet aggregation. So the chemicals which is released as a part of activation of platelets, mainly the ADP and platelet activating factor, further facilitate the platelet aggregation. Thus, accumulation of more platelets at the site of vascular injury facilitates the process of temporary hemostatic plug, that is a platelet thrombus formation. Then the formation of clot is initiated. Fibrinogen, glycoprotein 2B, 3A and thrombin promote the platelet aggregation. As a result of an injury, the platelets are activated simultaneously, and the activated platelets release the chemicals from their granules, especially the ADP and platelet activating factor, which further facilitates the platelet aggregation and forms a platelet thrombus. From there, it is the formation of the clot is initiated. You can see a diagram here, the role of platelet in hemostasis. A, B, C is given. First one is platelet adhesion. This first A is the platelet adhesion. What is happening here? What is all about platelet adhesion at the site of vascular injury, and you can see here the exposed collagen in damaged endothelium here. So as a part of vascular injury, the collagen is exposed in the damaged endothelium. So it promotes the adhesion. You can see the second one, what happens is platelet aggregation. So the second one is platelet aggregation. So you can see here platelet is aggregating here. Okay, the first one is the injury. There is exposure of the collagen in the damaged endothelium. The second you can see platelet aggregation. The third diagram you can see the C, what is formation of a fibrin threads around the aggregated platelets. So you can see just can you see the mouse here? What I'm pointing this where there is an injury here. When the injury is happening, there is an exposure of collagen in the damaged endothelium. So there is a site of injury. You can see site of injury arrow it is marked. The second one is endothelium exposed to collagen. You can see green color. That is exposed to collagen in the damaged endothelium. So there what happens here is you can see the platelet adhesion taking place at the site of injury. The platelets are adhered. This red ones are the platelets. The green is exposed to collagen. This is the site of injury. You can see a gap here. So these platelets are the platelets adhesion takes place in the site of injury. The second picture you can see the platelets get aggregated. See they are like a grapes. You can see it's like aggregated here. So a platelets are getting aggregated here, which forms the platelet plug. See the last one. The platelets aggregated here is what happens. It is surrounded, or you can see the blue mark here, which is fibrin thread. So like a thread it is formed. The fibrin threads in and around the aggregated platelets can be formed, can be seen here in third stage, that is a definitive plug formation. So there later a clot, you can see a fibrin thread itself is formed here, that is the end of the hemostasis. So there is a definitive hemostatic plug or blood coagulation have taken place here at the end of this third diagram you can see. So this is about the picture, platelet adhesion, platelet aggregation and platelet, the formation of the fibrin thread in platelet plug. So these are the role of platelets mainly. Similarly, this is one more diagram. You can see the synthesis of thromboxane A2 that promotes platelet aggregation. There is one more thing happening in platelet aggregation process. There is nothing but the thromboxane A2 is synthesized. Membrane phospholipids, arachidonic acid. You can see phospholipases and cyclooxygenase. Inhibition by steroids in this process. In this step, inhibition by aspirin, other NSAID. You can see prostaglandin G2, prostaglandin S2 with the help of thromboxane synthase, it converts to thromboxane A2 and results in promotes in platelet aggregation. So here NSAID is a painkillers which usually we use non-steroidal and inflammatory drugs. So cyclooxygenase inhibition by aspirin, other NSAID, phospholipase is inhibition by steroids. So you can see here at last the thromboxane A2 is synthesized, which can promote the platelet aggregation. So one more function of the platelet function was platelet activation and release reaction. Adhesion was over, aggregation was over. Now the last one is the activation and release. So adhesion of platelets to damaged vascular endothelium activates the platelets. Polymerization of microfibrillar actin occurs in activated platelets that results in pseudopodia or spicules formation. Platelets are also activated by tissue factors and chemicals released from the granules of the platelets, especially PAF. I told you ADP and PAF, platelet activating factor, are the chemicals secreted as a part of activation of platelets. Thrombin and ADP also promote activation. So again it is the same thing is coming. ADP and PAF are the main factors which is released as a part of activation. Even you can see thrombin also along with ADP and PF. So especially you have to remember the factor PAF, that's platelet activating factor. The activated platelets release various chemicals from their granules to the exterior via the canalicular system. The process is also called as release reaction or platelet release. That is release reaction and secretion. So release reaction, release means the chemicals are released, especially the ADP, PAF and thrombin, and there is a secretion, that is a platelet release is known as a secretion. So these are the different actions or the roles of platelets in the hemostasis. So this is a table showing the different factors that promote as well as inhibit aggregation and activation of the platelets. So factors promote aggregation and activation. You can see a list of factors: ADP, platelet activating factor, thrombin. As I told, these three factors already. You can see also serotonin, thrombospondin, fibrinogen, von Willebrand factor, immune complexes, plasmin, tissue plasminogen activator. Damage to the vascular endothelium directly can promote the aggregation, slowing of blood flow. So these are the different factors that can promote aggregation and activation. What are the factors that inhibit aggregation and activation? When the blood flow is very fast blood flow, prostacyclin, nitric oxide, or also known as EDRF. EDRF is the endothelium derived relaxing factor. Also, the inhibitors of thrombin generation and action can inhibit the aggregation and activation of the platelets.